everyone, my name is Aisha, and today we will be exploring the world of microbial metagenomics. First of all, what is microbial metagenomics? So, microbial metagenomics is the study of microorganisms in specific environmental samples by using functional gene screening. This process is what allows scientists to understand the functions of these microorganisms as well as understanding the interactions within microbes in their natural environments. Now you may be wondering, what are the functions of metagenomics? Byron, would you like to explain further?
Yeah, sure, Aisha. One of the functions is understanding microbial diversity. It helps us to discover and identify many different microbes in various environments. That means we can find out about microbes that we didn't even know existed before. Second of all, is to study microbial functions. We can learn what by studying their genetic materials. We can learn what they do and how they help in their ecosystems. For example, some of them break down waste, while the others, they produce essential nutrients. Besides, the third function is biotechnological applications. This research helps us to find out new enzymes and antibiotics for medicine and industry. For example, the new antibiotics can help in fighting resistant bacteria, and the new enzymes can improve manufacturing processes. In short, microbial metagenomics help us to find out more about microbes and use the knowledge on many beneficial purposes. That's all from me. Let's move on to having Anwar to speak more about the applications of metagenomics. Thank you.
Thank you, Brian. Now let me give you some information about the microbial metagenomic application. First, metagenomic helps in identifying microbial biomarkers associated with various health conditions. For example, specific patterns in gut microbiota composition have been linked to conditions such as obesity, inflammatory bowel diseases, IBD, and even mental health disorders like depression and anxiety. That is in the human health aspect. Second, I will speak about the environmental aspect. Metagenomics allows researchers to assess the biodiversity of microbial communities in different environments without the need for culturing individual species. This helps in understanding the composition, distribution, and dynamics of microbial population, which are crucial for ecosystem functioning. Finally, I would like to speak about the agriculture aspect, which is metagenomic analysis and identify microbes involved in nutrient cycling, nitrogen fixation, and phosphate solubilization. These insights are utilized in developing biofertilizer and biostimulants that enhance nutrient uptake, improve soil structure, and boost plant resilience to biotic and abiotic stresses. Now, I think it's time that we discuss the benefits and disadvantages of microbial metagenomics. Ayan and Brian, I'll pass it to you.
Thank you, Anwar. Now let's head straight to the main point. One of the benefits of metagenomic sequencing is that it is an upcoming research method which can be used to advance our study of microorganisms. It can detect rare and unexpected pathogens, which is why I think it should be utilized more in place of traditional methods. What do you think, I
think that's a good point, but these data analyses are very expensive and very complex, and it's hard for laboratories to conduct them on a regular basis. And if you're not skilled in the area, then you can get results that are full of errors.
That's true, but let's think about the benefits that metagenomic sequencing can provide. It has a higher sensitivity and it offers versatility, which makes it a worthy investment. And to add to that, it has a shorter turnaround time for the faster treatment of pneumonia, which can save lives.
That's a good point, but let's not forget that these sequencing technologies are only produce short read lengths, and they're more qualitative instead of quantitative. This can hinder the experiment's results.
Yes, but in exchange for that, we can acquire extensive information on the pathogens such as their origin, their strain level typing, and their genomes. Couldn't this crucial information be used to prevent outbreaks or epidemics?
That's a good question, but there are other factors that come into play, like environmental contamination and limited data referencing. These can hinder the experiment. I think there's a lot of issues that we need to discuss. Maybe Aisha and Elora could take a deeper look at it.
Such insightful information from both Ain and Brian. Let's now move on to the issues faced in microbial metagenomics. The first issue is that microorganisms have very complex living conditions. Some may vary in terms of temperature, pH, as well as pressure, whereas some may require more specific conditions, such as nutritional requirements. And some are even anaerobes, which would require extreme conditions that are difficult to replicate inside a lab. For the second issue, it is that the technical limitations, there are a lot of technical limitations that may be faced. A great example of this is metagenome assembled genomes, which would require the combination of short-read sequencing technologies and long-read sequencing technologies. This is due to short-read sequencing technologies being inhibited by repeat regions, which is why it would require long-read sequencing technologies to be able to cover these entire common repeat regions. So let's now pass it on to Elora, who will discuss more about these issues.
Thank you, Aisha. So moving on, micrometagenomics is both labor-intensive and resource-demanding, often missing specific microbes targets within the communities. And for experiments on microbes, on microbials, specialized labs are essential as some microbes thrive only in those harsh and anaerobic conditions. Moreover, acquiring costly equipment and expertise for precise data analysis is also a challenge due to the limited high quality tools that is needed. And for instance, like the UBLAST Kaiju, it has aimed to increase the speed of the metagenomics studies, which highlights the need for technical improvement for fast and more accurate data analysis. However, there's a need of high quality tools to do so. And additionally, reference database can also lead to the misinterpretation of the results. Besides that, it's also difficult task to obtain proper controls in many cases, especially in the clinical trials, as establishing proper controls would be crucial due to microbial composition variations that is influenced by the gender, diet, ethnicity, and genotype, and several other lifestyle factors. Moreover, proper controls are necessary to identify whether the signal is genuine and is not spurious or false results. So that wraps up our video presentation, and thank you.